Hello, we're glad you've joined us for this live webinar, From Research to Routine, Developing and Delivering Multiplexed Protein Mass Spectrometry-Based Tests of Clinical Utility. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning, and made possible by an open educational grant from Agilent Technologies. Agilent Technologies has had no input in the selection of speakers, content, or mode of presentation. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I now present today's speaker, Dr. Stephen Pennington. Steve is Professor of Proteomics at University College Dublin, Ireland, a chemist, Imperial College London, and PhD biochemist, University of Cambridge by training. Through engagement in a series of collaborations and international consortia, his current biomedical proteomics research is directed to the discovery and development of protein biomarkers. Ongoing projects are focused on the mass spectrometry-based measurement and evaluation of protein biomarkers in oncology and inflammatory disease to progress them to future clinical diagnostic utility. Steve's team is also investigating the mechanisms of disease progression, focusing on prostate cancer and psoriatic arthritis. Steve is highly energetic and enthusiastic with an innovative, open, and inclusive approach to his work. He has given over 200 invited presentations at international events, published over 80 scientific papers, edited and contributed to several books, including the editing one of the first books on proteomics that was translated into Chinese and Japanese. Steve has held roles as a committee member in the British Society for Proteome Research, currently Vice President, and the European Proteomics Association. He served as the lead organizer of the recent annual World Congress of the Human Proteome Organization, HUPO, held in Dublin. Recently, Steve founded the UCD spin-out company, Atturos, in December 2016 to support the delivery of OC Pro DX to clinical use. OC Pro DX is a prostate cancer blood-based biomarker test that has the capability to distinguish patients with organ-confined and non-organ-confined disease. The test will help inform men diagnosed with prostate cancer whether they should go on or stay on active surveillance. The Atturos team has the expertise and experience to deliver this and other multiplexed protein biomarker tests to market. Dr. Pennington will now begin his presentation.
Thank you, Judy, for that very kind introduction, and to colleagues in LabRoots and Agilent for making this webinar possible. As we've heard, I'm going to talk about taking biomarkers from research to routine and how we might do that by developing and delivering multiplexed protein mass spectrometry-based tests of clinical utility. I've just put up this slide now to acknowledge the funding agencies that have supported the work that we've been doing, from the Health Research Board and the Irish Cancer Society to more recently Enterprise Ireland. You've heard how I began life as a chemist, and as a chemist at Imperial College, there were two things that really put me off continuing as a chemist. They were NMR and mass spectrometry, and I was taught mass spectrometry by one of the world leading experts at the time, an individual called Howard Morris, who'd invented his own technique of mass spectrometry. He'd used that technique of mass spectrometry to work with John Hughes to identify the enkephalin pentapeptides, and that was a paper that they published in Nature in 1975. So having decided that I couldn't get to grips with mass spectrometry and NMR, I then went on to do biochemistry at Imperial, where I got involved in separating proteins by using 2D gels. So you can imagine my surprise and slight dismay in the early to mid-90s when it became apparent that if we were going to study proteins and separate them by 2D gels, mass spectrometry was going to be essential for their identification. And so, as we've already heard, I then had the opportunity to edit a book on proteomics, which was one of the first books on proteomics. In the intervening over 20 years now, we've been trying to use proteomics to understand disease mechanisms and identify disease markers. And increasingly, like many others, I've got frustrated at our lack of success of converting those mechanisms and markers to something of clinical utility. Particularly in the field of biomarkers, that frustration hasn't been just my own, it's been shared by others, and recently the MRC undertook a review of biomarker progress, not just protein biomarkers, but DNA and other molecules, and so that review was processed. I think one of the opportunities that we thought we had to try and increase our success was to collaborate, to go from local work to global collaboration, and we were part of one of the first global action plans that Movember, a funding agency that supports men's health research, initiated a number of years ago. So I think that whilst undoubtedly collaboration can make a major impact, more recently we've been focused on how we might convert some of the research discoveries that we've made into things of utility. And for the purposes of doing that then, as we've heard, I founded the company Atturos. So that company was founded in 2014, just a few years ago, and was officially spun out of the university just at the end of last year, December 2016, with a focus on taking routine or making routine some of the tests that we developed in the research arena and applying those for clinical utility. Now, clinical utility can look like this. This is a form from a hospital in Dublin, taken in 2006. And what it shows is a form in which a clinician will tick a number of boxes to have a number of tests performed to try and understand better the patient's current diagnostic situation. And so in this case, a tick box is ticked, a blood sample is drawn, that blood sample is then taken to a laboratory, and then in the laboratory the test's performed and the result fed back to the clinician such that he or she is better able to make a decision for the patient. This is that form in 2016. If you return to the same hospital in 2006, if you return to the same hospital in 2016, 10 years on, you'll see that the form looks different. And indeed it is different because two hospital departments have merged and they've put both of their tests on one form. There's been a change of font and a change of color. But the reality is that there are no new biomarkers, there are no new tests on that form. So over the 10 years in which we've been excited about developments in genomics and proteomics and metabolomics, there are no new tick boxes on these forms that enable clinicians to make better informed decisions about their patients. We can see in the bottom right here that a number of the tests are for proteins in blood, and including, for example, PSA and AFP that was ticked on this form. And those are proteins in blood that indicate whether a patient may have a particular cancer. For AFP, it's hepatocellular carcinoma, and for PSA, it's prostate cancer. So I suppose in the years that have been intervening, and as part of our frustration, we've been asking ourselves the question of whether the protein biomarkers that we discover will ever be useful. And if that's going to be the case, then how we will proceed to getting them to be useful. And what I've done here is outline the process that we use for going through biomarker discovery and assay development and then subsequently confirmation and evaluation and validation. And there's nothing new on this slide. This just shows a version of a process that many people have adopted to try to get to biomarkers that may be of clinical value. Of course, the end point of all of this is a biomarker that will result in a clinical test. And I'm just trying to draw an arrow here just to show and highlight that that's a clinical test that will be the output of all of this work. I've separated this process into a number of discrete phases in which we begin with discovery to identify some protein biomarker candidates. We can confirm those protein biomarker candidates by developing an assay to them. And increasingly now we realize that not individual proteins will form a biomarker, but it may be a panel of multiple proteins. And so having developed assays to measure a panel of proteins, we can evaluate the performance of that panel of proteins and ultimately have a clinical test. I've drawn the analogy that this is a bit like climbing Everest, that we go through the process in multiple stages, often reaching camp 1 and then having to decline again to base camp so that we can reacclimatize. Eventually, we progress through camp 1, 2, 3, and 4 and ultimately reach the summit of Everest. And of course, when we get to Everest, we can view the top of the world. So we've begun at base camp and even got maybe to camp 1 or 2, and the result of that has been a whole series of biomarker discovery projects in a range of different diseases in which we've attempted to identify biomarkers from tissues or cells, from serum, various biological fluids. And the result of all of that work really has been summarized in a recent review back in 2011, in which George Post described all of this as biomarker futility. Biomarker futility because the omics haven't delivered any new biomarkers to the clinic. And George Post identified what he thought were a number of reasons as to why that's the case, including the fact that we were using or have been using poor specimens, that we've had a fragmented approach to the way that we do this. So I'm getting a little carried away with the animations there. So there's no doubt that the reasons that George has identified have indeed prevented us from getting to effectively to the Everest of biomarker discovery, because the Everest of biomarker discovery would be a new clinical test. Protein biomarkers discovered in our laboratory that were now incorporated into a clinical test that was used by patients by physicians for patient benefit. And of course, we'd ideally be at the peak of the summit of Everest. And so I have to confess that I'd like to reach produce a new clinical test, effectively to reach this Everest. And there's a little bit of sibling rivalry going on here, because I have a younger sister who has reached the summit of Everest twice, the real Everest, not the biomarker Everest, and she's done that twice. She's the only UK woman to have summited Everest twice, and I'd really, as I say, in a sense of sibling rivalry, like to be able to beat her by summiting the biomarker equivalent of Everest, that is to develop a new clinical test. And if we're going to do that, you could obviously ask yourself, how are we going to be able to do it? Can we discover biomarkers and develop them into new tests of clinical utility and value? And many of the challenges associated with doing that are really well rehearsed now. And as well as in the Post review that I've just mentioned there are a number of articles that have sort of outlined some of the challenges faced in taking a biomarker that's been discovered and converting it to a new clinical test. So there are undoubtedly very many challenges in that process, and that perhaps in part explains why very few, if any, have reached the Everest of biomarker discovery and development. So you can imagine my surprise a few years ago when at a biomarker meeting, a young bright-eyed consultant, Dean Griffiths, said actually to get a biomarker to clinical utility is simple. He said all that you need to do is identify a need, find the solution, and then scale it. And of course, in those terms, it is very straightforward, and those are just the discrete steps that you need to go through. And I suppose back in 2014, when I first heard this, that got us thinking to what we really need to do is to begin the process at the end. That is, that we need to identify the clinical test that we would wish to develop, and then begin the process of discovery. And so just to reiterate, that's to identify the clinical need and the clinical test that will address that need, and then begin the discovery process. And I suppose since around 2012, 2014, when we were formulating these ideas, that's when we began studies in two disease areas. And I just want to illustrate one of those disease areas and really begin by illustrating that beginning at the end process. And so the disease area is prostate cancer. So I'm sure many of you will be aware of the impact that prostate cancer has. But what you may not be aware is that 1 in 6 men get diagnosed with prostate cancer at some time in their lives. This is in the Western and the developed world. So in Ireland, which is a small country, that's over 3,500 men a year. In the UK, it's over 30,000 men. In Europe, it's close on 500,000 men every year get diagnosed with prostate cancer. And that diagnosis is made initially on the basis of a protein, PSA, that's found in blood. And so PSA is a small protein found in blood, and an elevated level of that protein is suggestive of prostate cancer. Once it's found to be elevated, the clinician then will perform a digital rectal exam, a DRE. And the DRE is what it really says on the tin. That's where a gloved digit is inserted into the rectum and the prostate gland palpated for any abnormalities. If abnormalities are felt, then the patient is then subjected to a biopsy. And the biopsy is where a biopsy needle is inserted into the prostate, guided by ultrasound, and most commonly, until fairly recently, this was done by transrectal. More recently, it's transperitoneal. The transrectal biopsies carry with them a risk of infection, and so the patient very often will have to have prophylactic antibiotics, and because it is so painful, will have to have significant anesthesia before the biopsy procedure. Once the biopsy tissue is removed, it's then sectioned and scored by an expert pathologist according to a Gleason grading system from 1 to 5. So armed with an elevated PSA, an abnormal DRE, and a Gleason score of the tissue, the clinician is able to make a diagnosis of the disease. So if you imagine, for example, this is a young man with his back to us. It's Harry, young. He's in his mid-40s. He has a routine health check because, although there's nothing wrong with him, his work suggests that it would be a good idea for him to have a routine health check, and he does that. And it comes back that his PSA is elevated. It's actually quite elevated. It's at 12.2 nanograms per mil, where the threshold for suggestive of possible prostate cancer is 4 nanograms per mil. Subsequently then, Harry has a digital rectal exam, and that comes back as being slightly abnormal. Subsequently, after this, then he has a biopsy, and his biopsy comes back as Gleason 3+4. That's 2 regions have been scored, and that's a Gleason 7. The result of this is unambiguous. Harry has prostate cancer. So recall, a little while ago, Harry was asymptomatic, but just went for a routine test, and now, a week later, it's absolutely clear that he has prostate cancer. What to do next is determined by Harry's risk score. And in his case, he's between low and intermediate risk, regardless of whether he's in Europe or America. And that will then determine what will follow his diagnosis. And what follows his diagnosis is that he clearly has to make, with his clinician, a very significant, and it's a life-changing decision. The decision is to either have active and invasive treatment of the disease, that is a radical prostatectomy where the prostate gland is removed, or radiation treatment where the prostate will be treated in situ with radiation. Both of those treatments carry with them significant comorbidities. Harry is likely to experience urinary dysfunction and bowel dysfunction, as well as erectile dysfunction, all of which will have a major impact on his quality of life. He's also likely to experience the side effects of hormone therapy, which include some feminization. The third option that Harry might have would be to go on active surveillance. That's where the disease is monitored until it's felt that treatment would be absolutely necessary. And active surveillance will involve repeat PSA testing, some imaging of the prostate potentially, and possibly repeat biopsies. The main comorbidity associated with active surveillance is one of anxiety for Harry to have to live with knowing that he has prostate cancer. And so he's likely to come under significant external pressure to have his tumour treated. Until very recently, it was really the majority of men with Harry's kind of diagnosis of prostate cancer, with those molecular and clinical features, would have had the first 2 options. They would have had either a radical prostatectomy or radiation treatment. However, at the end of last year, published in New England Journal of Medicine, was a paper that showed the outcome of a clinical trial in which over 500 men were randomized to those 3 treatment options, the treatment options being radical prostatectomy, radiotherapy, or active surveillance. And those men were followed for 10 years. And 10 years later, it became apparent that there is no difference in deaths from prostate cancer among the three groups. So that means that 2/3 of the men had this invasive treatment that has significant life-affecting comorbidities without it having any impact on their death from prostate cancer. And that's led to the realization that there is massive overtreatment of men with prostate cancer, and the realization also that less is more. That for those men who are chosen well, active surveillance is perfectly safe. Indeed, it would be the preferred option because the quality of life is significantly improved for men who don't have those invasive treatments. However, the current tools for selecting which patients to go on active surveillance are inadequate and imprecise. And indeed, there are no minimally invasive blood tests that support the decision to go on and stay on active surveillance. Because it's recognized that men who have multiple biopsies, because they're so painful, so uncomfortable, and actually carry with them significant risks themselves, that most men will choose after a number of biopsies to then just have one of those active treatments. There are also significant imprecisions associated with the biopsy assessment process. So a number of years ago, we undertook a roadmap of decision-making as men are treated with prostate cancer. And the urologists that we work with suggested that this second question of knowing whether the disease is organ-confined or non-organ confined would be a significant question to be able to answer and would help in the process of making decisions for men diagnosed with prostate cancer. So just the next slide just illustrates that in organ-confined disease, the tumour, allow me just to highlight it, the tumour is resident within the prostate, obviously grows over time. In this case, it may, as is often the case with prostate cancer, have multiple foci until eventually it becomes non-organ confined, that is, that it spreads into the seminal vesicles and into the systemic blood system. So the difference then between an organ-confined disease versus a non-organ confined disease is that in the case of the non-organ confined disease, it's likely to become ultimately metastatic and be life-threatening, whereas organ-confined disease will never be life-threatening. So armed with that piece of information and that desire, we began the discovery process. And in this case, the discovery process began with taking serum samples from men who either had organ-confined disease or non-organ confined disease and subjecting that to standard label-free LC-MS proteomics discovery. And in this proteomics discovery workflow, we took the serum samples, depleted them of abundant proteins, digested the proteins with trypsin, cleaned them up, put them onto mass spectrometers, in this case Q-exactive and Q-TOF mass spectrometers, and undertook qualitative and quantitative analysis, and then used a series of statistical tools in a range of softwares available to identify proteins that would potentially discriminate between organ-confined and non-organ-confined disease. And through this process, we identified a series of candidates. And these candidates were also then supplemented with discovery experiments that we did by gels and by reviewing the literature. So that ultimately we ended up with 64 candidate proteins. So these are proteins that may discriminate between organ-confined and non-organ-confined disease. Now, in the past, armed with a biomarker candidate like this, we would have had to have selected one or two of the best candidate proteins and progressed them through the pipeline that I showed earlier. What we have now been able to do with the advent of multiple reaction monitoring is not develop assays to a single of those proteins, but to develop assays potentially to all of the proteins. So multiple monitoring mass spectrometry, MRM, multiple reaction monitoring, is a process in which we can target the measurement of specific proteins. And so just to outline the process, if in a mixture of proteins there's a protein of interest that we wish to be able to measure, we can identify peptides that for that protein that are target proteins, peptides that we're going to measure. And these peptides have unique mass to charge. The mass to charge ratio of the peptides derived from this protein is such that the peptides can only have ever come from our protein of interest and not from the other proteins in the complex mixture. So having identified triptych peptides that are unique to the protein of interest, we then measure these in a triple quad mass spectrometer. So depicted below is a triple quad mass spectrometer in which all of the peptides, all of the ionized peptides, move into the first quadrupole. And we select a mass filter in that first quadrupole that only allows the peptide of mass to charge ratio of interest to us through into the second quadrupole. The second quadrupole acts as a collision cell, and so that peptide ion is fragmented. And of course, if the peptide is the peptide we think it is, we know the masses of the fragments that are generated. And so in the third quadrupole, we can select a filter that will only allow fragments of a size that we predetermine to ultimately reach the detector. And so what we can see here is a retention time trace showing, in this case, the intensity of the fragment that is of the highest intensity from our peptide. If we want to be sure that the peptide that we think we're measuring is indeed that peptide from this complex mixture, we can, of course, in the third quadrupole, select a second fragment ion mass of interest and measure the intensity of that fragment ion, and subsequently a third fragment ion of interest and measure the intensity of that fragment ion. And we can keep doing that process for as many fragment ions as we wish to use to confirm the identity of the original peptide. So this method won the Method of the Year from Nature a number of years ago, and at the time I wasn't even aware that Nature ran a Method of the Year competition. And there are many good reasons why targeted proteomics by multiple reaction monitoring won that particular method, that particular accolade. And that's because it has a dynamic range of over 4 orders of magnitude, which is probably greater than any other analytical technique, certainly that I'm aware of. That you can multiplex it very easily, and so we routinely measure over 50 proteins per assay. It can be quantitative. So by incorporating peptides of known amount into the assay, you can measure the fragment ions from those peptides and compare them with the fragment ions of the peptide of interest. The trick, of course, is to incorporate peptides that are the same as the peptide of interest, except that they are isotopically labeled. They may have 15N carbon or 15N nitrogen or 13C carbon incorporated into them rather than the natural isotope. Triple quad mass spectrometry can be very robust, and on many occasions these assays can be developed to CVs of less than 10%, and I'm going to illustrate that in a moment. However, as with many methodologies, there are limitations, and in many cases this approach is not as sensitive as an ELISA. But there are ways that can be used to circumvent that limitation. So just to reiterate, the method requires that we identify and measure a peptide that's unique to the protein of interest, and we measure that peptide, its mass to charge ratio, and fragments of it that are generated in the mass spectrometer, in this case a triple quad mass spectrometer, a very robust mass spectrometer. So we developed our first multiplexed assay in 2006, and that was an assay to cytochrome P450s. In this case, 16 cytochrome P450s measured simultaneously in around 30 to 40 minutes. So that's like doing 16 quantitative Western blots or quantitative ELISAs simultaneously on one individual sample. Subsequently, that approach has been patented and is now available as a product from Sigma. So that just illustrates, I think, how robust these assays can be. We developed subsequently an assay to 48 proteins and published that a few years later with colleagues from Agilent. In this case, Christine Miller from Agilent published that paper with us. So over the 10 years that we've been doing this, we've now developed a very robust Mrm workflow in which from a candidate list we can pick peptides and prioritize those peptides. And we found that it's very helpful in going through this process to use Spectrum Mill from Agilent. It's a very robust approach for identifying peptides, and the peptides that are prioritized by Peptide Selector in our hands turn out to be ultimately very good peptides for measuring the proteins by MRM. Initially, we begin an unscheduled analysis of the peptide to identify its retention time and the reproducibility of our methods. And then once we know the retention time of the peptide, we can target the mass spectrometer or program the mass spectrometer to just look for the peptide that we're interested in during a specific time window, and we call that a scheduled method. So then, having a scheduled method to a peptide of interest, we can optimize the method by optimizing the collision energy and doing that through the analysis of synthetic peptides. So there are a whole series of steps that we can go through to optimize this workflow. So going back to our 64 candidate proteins that we discovered that have the potential to discriminate between organ-confined and non-organ-confined disease, we were able to develop MRMs to those 64 candidate proteins. In fact, it turned out that we were able to develop MRMs to 63 of the 64 candidate proteins for doing biomarker evaluation. And obviously that targeted approach now is much more effective than attempting to develop assays using antibodies to those individual proteins. So we were able to develop multiplexed MRM strategy for measuring those candidate proteins and use additional patient samples to evaluate them. Of course, the devil is in the detail, and what I'm showing here is over 30 biomarker MRMs for some of those candidate proteins. And so these are really carefully selected and optimized MRMs for those some of those candidate proteins. And I think that this is perhaps one of the most challenging parts of this process, is developing really robust assays that work in the particular sample that's being used. And in this case, it's a crude serum sample. So now, what I've highlighted here is that using this targeted approach, we can confirm whether the candidates do indeed increase from our discovery cohort of patients, and we can then use the targeted approach, this MRM approach, to evaluate the clinical performance of a panel of proteins, and I would say then move us 2 steps further towards developing a clinical test. So we're perhaps reaching Camp 3 on our climb up Everest to a clinical test. When we did that for the first 31 of those candidate proteins, and so what this is showing is the MRM measurement of 31 of those candidate proteins in a different patient sample set, we can measure the area under the curve for their ability to discriminate organ-confined from non-organ confined disease. That's organ confined from non-organ confined disease, and that produced an AUC of around 0.8. The clinicians informed us that was a very valuable AUC, and it was certainly worth continuing. Our statisticians confirmed for us that this wasn't by random chance, so they tested this for randomness and were able to show that those AUC values generated could not have been arrived at by random chance alone. And so on the basis of that, the university filed a patent on those prostate cancer biomarkers, and that was in December of 2013. We then developed the assay further and as I said before, moved that to 63 proteins, so measuring 63 of the 64 candidate proteins in a separate cohort of patients, again a modest-sized cohort of patients, and using a range of different statistical approaches. Again, the AUC values were around about 0.8. So I suppose that's given us sufficient confidence that we have identified some proteins that can discriminate organ-confined from non-organ-confined disease, and that we may have a solution to the need that we'd identified. And so the solution in this case is a multiplexed protein blood test that can discriminate organ-confined from non-organ-confined disease, that begins by taking a blood sample, making serum, the serum being digested with trypsin, and the peptides going onto a triple quad mass spectrometer to perform this test, and the test then being used to generate a confinement score, the probability with which the prostate cancer is confined to the prostate or has spread beyond it. And as I've indicated, the university filed a patent on that panel of prostate cancer biomarkers. So that's all very well to have a panel of proteins that seem to have the ability to discriminate between organ-confined and non-organ-confined disease. But if it's going to become a clinical test, then what we have to demonstrate is both the analytical validity and the clinical utility of that test. And the analytical validity means that we can perform the test routinely and robustly, sufficiently robustly that it could be used to diagnose and make a treatment decision for a patient. And so what we did a number of years ago was to set up a small laboratory in which we simply take samples that are stored in a -80 freezer, digest them with trypsin on a workflow, and then put them into a triple quad mass spectrometer. And all that happens in the laboratory is those simple steps, and everything is done by standard operating procedures. So this really is to test the analytical robustness of the methodology. We incorporate into this a lot of quality control samples, and one of the quality control samples that we include is a peptide mix. We call it the TSPA peptide mix. And this just is measuring the retention time of those TSPA peptides on the chromatography that precedes the mass spectrometry. And we see routinely that these peptides have a retention times of less than 2.5%. And that means that the peptides elute at the same time on every single occasion. And it means that we can then assess the peak areas more robustly and more automatically. And so when we try then to measure the analytical reproducibility of the test, and this is a busy slide, but essentially what it shows is it shows 4 different runs, one here, a second here, a third, sorry, there's a slight pause when we get to the tool, a third and a fourth as we move towards the right. 4 different batches run over 4 days, and those 4 batches have in them a number of QC samples. So wherever there's a shaded box, that's where a QC sample has been run. And so now within those QC samples, we've run 130 patient samples approximately. And we have both sample and analytical replicas. And this really is just to demonstrate the robustness of the methodology. If we now measure the area of those 16 QC peptides, we can see that the area of all of the QC peptides, the average CV over 160 samples, that is 96 hours of mass spec runtime, is around 6.5%. So that demonstrates the robustness of these particular assays for the QC peptides. If we now incorporate exogenous spiked peptides, that is where we have isotopically labeled peptides, and so now this controls for the loading of the mass spectrometer, and we ratio the endogenous to the exogenously spiked peptides, we see that those CVs can be brought to less than 5%. And so that means that the robustness, the analytical robustness of the assay is such that it could be a clinical diagnostic assay. And so the next slide just shows 99 peptides that are under evaluation for around 60 biomarker proteins that we're currently measuring. Again, over 96 hours of runtime. So that's over 4 days of mass spec runtime. And for all 99 peptides, the average CV of all of the peptides, not removing any of them that are have uncomfortably large CVs, so this is an unbiased assessment, it shows that they have a mean CV of 12%. Again, demonstrating that that's suitable for a clinical diagnostic assay. One of the questions that's often asked is that of course, these assays take time. Each of the assays on this occasion takes around 30 minutes, and they're run serially, one sample after the other. So what we did on one occasion was just to count up the number of samples that we'd run in that small laboratory with a single triple quad mass spectrometer over a period of 28 days. And this was just a standard 28-day period in that laboratory. And we can see that the total number of samples run on a single mass spectrometer was around 1,600 samples. So that's 1,600 samples, 1,300 of those were patient samples, and around 300 of them were quality control samples. And so that demonstrates, I think, that for one instrument, we could implement this assay for all of the prostate cancer patients in Ireland. It gives, I suppose, a sense of the scale and the realization that the throughput of a mass spectrometer, depending on the assay and the clinical need of that assay, means that it could reliably be used to measure and assess prostate cancer patients in this case. So the stage we're at the moment is to have taken that study further and subject it to an ongoing evaluation that's been funded by Enterprise Ireland, in which we're measuring the panel of at least 63 proteins in 1,500 patient samples, evaluating the analytical performance further and being able to assess rigorously the cost and the throughput of that assay. And then I suppose the very next stage, which is to evaluate the commercial potential of the assay. Indeed, how, if we develop this assay and it shows that it's analytically and clinically robust, whether it can be brought to patient use. And the reality is that to bring it to patient use, it would need to be commercialized. And so we need to understand who's going to do that, who's going to pay for the test, what regulatory path it may have to go through, and ultimately what healthcare impact it would be likely to have. And this is where we have spun out Atturos to be able to do that for the OC ProDX test. So that's really to be able to bring this OC ProDX test, as we call it, to clinical use, to take it to those next stages. Of course, we have the opportunity then, because this is a very flexible mass spectrometry platform, to develop other tests. And in the consideration for doing that, and funded through a Horizon 2020 EU business development grant, we've established a business model in which the patient sample is taken either in a hospital or a phlebotomy department, and that sample then is sent to the laboratory, in this case the Atturos laboratory, where the test is performed on a triple quad mass spectrometer, and then the score, which is obtained through a proprietary algorithm, is sent back to the patient and the physician. And so the testing is done in a centralized laboratory. And so in the last few weeks, we've started the process of developing that Atturos centralized laboratory. And this just shows a picture of the laboratory as it stands at the moment, about 12 weeks on from its development. And you can see that this is just a standard mass spectrometry laboratory in which we have a number of triple quad mass spectrometers. There's two in this picture, and there are now, as of Monday last week, 3 triple quad mass spectrometers to be able to continue the development of that workflow. One of the questions we often get asked in developing this process is whether these MRM assays give the same results as ELISAs. So whether if we were to measure a protein by MRM, whether it would give the same result as a clinical test performed in a clinical diagnostics laboratory. And so that is whether MRM is equivalent to ELISA. We could spend a significant amount of time debating whether that's absolutely necessary, but it does seem to be a consideration that most people, or a lot of people, place as being one of importance. I'm not sure I share that view, but nonetheless, we were recently asked to demonstrate it. And so in that new laboratory, we developed an MRM to a protein called C-reactive protein, and we did that because we had ELISA measurements that had been undertaken on a Roche diagnostics platform in a series of patient samples from patients not now with prostate cancer, but patients with psoriatic arthritis or rheumatoid arthritis. And so this just shows the results of a very recent experiment in which an MRM assay to CRP was developed over a 3-day period, and that MRM then used to measure CRP in 60 patient samples, and that data compared with the data from ELISAs of those same 60 patient samples. And I think you can just about see here that the correlation coefficients for the MRM measurement and the ELISA measurement are very similar indeed. And indeed, when we look at the individual patient samples and compare the PSA patients with the RA patients, whether it's by ELISA or by MRM, we see that we get very similar statistically increased CRP in the RA patients compared to the PSA patients. So I think the value in this kind of data shows that in some cases, and indeed in this case, the MRM is entirely equivalent to an ELISA. The difference, of course, is that this MRM took 3 days to develop compared to the time taken to develop an ELISA. And of course, what we're able to do with the MRM is to multiplex it much more effectively. And so I think then to summarize, and I hope that's illustrated a process that we've gone through to try to develop a multiplex protein test for prostate cancer, and we have a number of tests that we're developing. And so if we want to take biomarkers from something we've discovered in the research laboratory to a clinical test, to a diagnostic, then I think it's evident that the biomarker should be fit for the purpose for which we intend it. And of course, we need to know that purpose. And this reiterates the start at the end principle. The MRC, in reviewing the process of generating diagnostics from biomarkers, said that there needed to be a process of reforming the regulatory review process. And one of the reasons we've set up the Atturos laboratory is that at the moment these tests can be undertaken in a central laboratory if the sample is sent to that laboratory, as opposed to the test being distributed to hospital laboratories. The MRC suggested that the reimbursement of these tests needed to be increased. And clearly these are not cheap tests necessarily, but their value lies in the savings that they provide to the healthcare systems. They suggested that there needed to be increased investment in research to identify better biomarkers moving forward. There needed to be increased rigor for the assessment of biomarkers, including increased rigor in the publication of biomarkers. And one of George Post's major criticisms was that many of the publications that have supposedly led to reported the identification of new biomarkers have not themselves been sufficiently robust. And then, of course, ultimately, we need to adhere to a high-level evidence base if we're going to recommend the use of these biomarkers for making clinical decisions. And that really means that the tests have to have analytical validity as well as clinical value. And of course, they also are going to be required to have financial value if they're going to be brought to market. So I think maybe a recipe for success, and this is purposefully a question mark because the jury, I think, is still out on this, is that we need to identify a clinical need. We have to start at the end. We then need to conceive a product to fulfill that clinical need, and that's really important. In our case, once the product was in for patients with prostate cancer, that defined the required throughput and the speed with which the test needed to be performed. And that really does then revolve around designing the test to be fit for purpose. Whether it's going to be point of care or it's undertaken in a clinical laboratory, it'll define the turnaround time and the throughput required. And then we need to undertake a process of evaluating the true clinical value of the test that we think we've developed. And in our case, that's something that's very much ongoing. And once perhaps that's been done, or maybe even before it's been done, we need to identify the monetary value of the test and the reward and the route to which it's going to be achieved. And some would argue that once all of that has been theoretically undertaken, then you can begin the process. I hope what I've been able to illustrate in the brief time that I presented this presentation is that I firmly believe that targeted proteomics methods, these MRM methods, have utility for clinical tests, that ultimately there will be clinical tests that developed that require MRM measurement of multiple proteins. It remains for me to acknowledge the many individuals associated with the work that we've been doing, including some from two individuals identified from Agilent Technologies. There are many more members of my group. But of course, most importantly, the patients who've provided the samples to enable us to do the discovery and the evaluation studies, and a whole team of individuals. Ultimately, we want to, as we've indicated, move these tests forward through Atturos, the small company that we've spun out. And I suppose the vision of the company is to deliver diagnostic tests of global impact, tests that improve outcomes for patients and a better financial situation for healthcare systems, that is a saving for healthcare systems. So with that, I'll end, and I'll be very happy to take any questions that you may have. Thank you.
Thank you, Dr. Pennington, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Pennington will answer as many questions as time permits. And the first question is it necessary to measure multiple transitions per peptide? And also, doesn't quantification work with the most intense transition?
Yeah, thank you for that question. It's a good question. So quantification does work with the most intense transition, and indeed there are a number of papers that show that if you measure the most intense transition versus measuring the three highest intensity transitions, the quantification data is very similar. So quantification is done with the most intense transition. However, we measure multiple transitions per peptide because those transitions, and the rank order of the intensity of those transitions allows us to determine whether the peptide and the fragments generated from it are specific to the peptide that we're attempting to measure. So they provide, if you like, a quality assessment that the transitions that we're measuring do indeed come from the peptide that we're attempting to measure.
Do you include isotope-labeled peptides for each peptide you're measuring?
At the moment, we don't, and I think there's ongoing debate about whether this is really necessary. In many instances, in equivalent scenarios where people are measuring, for example, multiple messenger RNAs using reverse transcriptase PCR, they don't absolutely quantify each of those transcripts. Instead, they measure the relative levels of the transcripts and use an algorithm to derive a score or a numerical value for the combination of them. And I think that's something that we anticipate will be done in a similar fashion here, that we will measure the relative amounts of those peptides and then using the algorithm that we've developed produce a score. And it won't be necessary to analytically measure and quantify each of the peptides, and therefore we don't include isotope-labeled peptides.
Is the test specific to prostate cancer? Have you tested it against other cancers?
Again, that's an interesting question, and it's one that we've given significant consideration to, because what the test isn't is a test that's used for the diagnosis of prostate cancer. It's a test that's used subsequent to a gold standard diagnosis of prostate cancer, which is a tissue biopsy. So we know that the test is only going to be used in patients who have a diagnosis of prostate cancer, and therefore we haven't tested whether it measures or it is interfered with by other cancers, because we're assuming, because the men that have been diagnosed with prostate cancer have been very carefully clinically evaluated, that these are men who have prostate cancer and not other tumors.
Does the test work on plasma?
Again, a good question, and this really revolves around the difference between plasma and serum, and the challenges in using both of those blood matrices. At the moment, we are beginning a process to evaluate whether the test works on plasma. There's a priori no reason why it shouldn't, but for considerations that I probably don't have time to go into, our preferred matrix has always been serum. That, as with all of these things, when you choose something, it has advantages and limitations. And of course, the biggest limitation with serum is that it requires a blood draw and then subsequent centrifugation, which limits then the point of use of the test. So in this case, the sample that's used for the test has to be taken in a clinic that has access to the ability to prepare serum. And so, for example, using serum means that it could never be a test, at the moment, couldn't be a test that could be done with a blood draw at home, whereas there's a possibility that would be the case if it worked on plasma. So at the moment, we're evaluating its ability to work on plasma, because that would increase the number of sites at which the blood could be drawn for use for the test.
How efficient is expression of a particular biomarker in diagnosis of a specific cancer, and can a single biomarker be found in different type carcinomas?
So I suppose that goes back to my question about diagnosis. And so what we specifically haven't attempted to do here is to identify a protein that could be used to diagnose the cancer. I personally think it's highly unlikely that a single biomarker will be found that's specific for particular cancers, and that means that because there are a whole series of considerations, the expectation would be that protein would have to be cancer specific. It would have to be a protein that's produced in the cancer, is secreted or finds its way into the blood, and then could be measured at the level required to be able to detect the tumor at an early stage. And I think that's a tall order for a single protein. It's probably even a tall order for a number of different proteins. And of course, if you're going to evaluate then whether the protein or proteins really are specific for a particular cancer, then the evaluation studies that need to be performed need to rule out the possibility that that protein also identifies patients with other tumors. And so the samples then need to come from patients who've got a range of different tumors, as well as obviously healthy, normal controls. And that means that that whole process of identifying a protein that's a diagnostic biomarker for a particular cancer is significantly more challenging.
As we're waiting for additional questions to come in, I would like to once again thank Dr. Pennington for his presentation. Do you have any final comments?
Sorry, do I have any final comments,
yeah.
Judy?
Do you have any final thoughts you'd like to share?
I suppose my final thought that well, a couple of thoughts. The first is that I think it's becoming apparent that these multiplexed MRM measurements are being converted into tests, that's occurring as with many of these molecular tests. Most advances being made in the area of cancer, and so there are MRM tests being developed for lung cancer. The prospect of these multiplex tests being used as laboratory-developed tests, I think, has been improved significantly by a recent ruling from the FDA saying that multiplexed mass spectrometry-based tests will be subject to 510K exemption, and so that means that for the regulatory process they can be advanced. And so I think that the interest in developing multiplex tests for cancers is significant. In our own case, we're also developing these, as alluded to previously, in psoriatic arthritis and inflammatory conditions and a number of other disease states. So I think they hold enormous potential for the future.
Thank you. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our educational sponsor, Agilent Technologies, for today's webcast. This webcast can be viewed on demand through February 2018. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.